Good morning, my dear friends. In this lecture, we are going to discuss regarding grainage, chalky rearing, and late age rearing. Quality seed is the backbone of sericulture industry. Such quality seeds are produced at breeding centers, grainages, and distributed to the farmers. In India, usually three-tier system of seed production is adopted, that is at P3 center, P2 center, and P1 center. At P3 center, basic or nucleus seeds are produced. At P2 center, foundation stock seeds are produced. P2 center also serves as the germplasm bank. At P1 center, this center receives the cocoon from the P2 centers, and the parents of commercial layings are mass multiplied and distributed to the rearers. Silkworm eggs are called as seeds or silk seeds. There are two types. That is one reproductive seeds and another one is the industrial or commercial seeds. The reproductive seeds are produced in very limited quantity in special breeding centers by technically qualified persons. The purpose of the production of the reproductive seed is for maintaining the racial purity and to produce the parent for the commercial seeds. The commercial seeds, on the other hand, are produced in very large quantity in special organization known as grainages. The purpose of the production of the commercial seed is to supply to the farmers. What do you mean by grainage? Grainage. Grainage is a center where the disease-free silkworm seeds are produced in large quantity under scientific line. This grainage center purchase cocoon from the certified seed cocoon produced. These centers purchase cocoon from the certified seed cocoon producers, allow them for moth emergence, and then make moth sexing and obtain large number of seeds. And these seeds are mass multiplied and distributed to the rearers. These grainages are mostly government owned and may also be owned by the licensed private owners. See here, plan of grainage building. A grainage building must and should contain the following facilities. That is one, a room for accommodating cocoons or pupa of different strains. A room for coupling and oviposition, incubation room, laboratory, egg processing room, cold storage room, dormitory, and office room. The location of the grainage. The grainage must be located in cooler areas of the locality, and a grainage must be situated in the center of the sericulturally active area. See here activities taking place at a grainage. The grainage center purchase cocoon from the certified seed cocoon producers. The cocoons are placed in well-ventilated room which are provided with proper temperature of 23 to 25 degrees Celsius and humidity is provided 70 to 80%. And here moth emergence is allowed. These grainage rooms may be kept dark and light may be supplied suddenly on the expected day of emergence to bring uniform moth emergence. These emerged moths are sexed and used for breeding purpose to produce seed eggs. 3 hours of mating can secure the maximum fertilized eggs. The sexed females are then made to lay eggs on paper sheet or cardboard which are coated with thick gummy substances. Egg sheets are disinfected with 2% formalin and then washed with water to remove traces of the formalin and then dried up under shades. The dried eggs are weighed to the required standard and packed in a small flat boxes with muslin covers and dispatched to the farmers. Now the silk seeds are in the hands of the farmer. Farmer will incubate them and obtain silkworm larvae. These larvae are provided with good quality food. Grown-up larvae mounted on the mountages. On mountages, these larvae form cocoons. Cocoons are harvested and marketed. Marketed cocoons undergoes processing, and from that processed cocoon, we extract silk. Now we are going to see all the steps in details. Egg incubation and hatching. Silkworm rearing starts with silkworm egg incubation. Eggs are incubated under optimum temperature of 25 degrees Celsius and humidity of 80 to 85%. Under these conditions, eggs are expected to hatch within 10 to 12 days period. See here, eggs are taken into the hatching tray for incubation purpose. A hatching tray can accommodate around 30,000 eggs. Here, hatching tray is covered with tissue paper. Over it, another plate is placed for proper coverage and it is placed for incubation. See here, several hatching trays are kept for incubation in a single large tray. Block boxing. To ensure the uniform egg hatching in silkworm larvae, the eggs at pinhead stage are kept under total darkness, either by wrapping them in black paper or keeping them in black box or dark room for 2 days before hatching. This process is known as black boxing. The next day they are exposed to diffuse light so that the larvae hatch uniformly in response to photoic stimuli. In silkworm, eggs generally hatch in 10 days, so block boxing is done on the 8th day or 2 days prior to the egg hatch so that 90% hatching can be obtained. See here, after block boxing, 90% hatched larvae on the tissue paper. Now we have larvae and tissue paper are on hatching tray. We have to transfer them to the new tray or rearing beds. Transferring of newly hatched larvae to new trays or rearing beds. There are two acceptable methods. This is one method and this is another method. In this method, we are covering the hatching trays with porous nylon net and providing chopped tender leaves as feed over this net. The hatched silkworm will crawl onto the chopped leaves and start feeding. Then take out the nylon net with crawled larvae and transfer to a new tray and give them first feed. Second method is brushing. The process of transferring the silkworm to rearing trays is known as brushing. The hatched worms are brushed with a feather from egg cord directly onto the rearing bed. During brushing, care must be taken not to injure the worms and give them first feed. Silkworm undergoes 4 moltings and gives 5 instars. Molting or egg dice is nothing but periodical process of shedding the old cuticle accompanied by the formation of the new cuticle is known as molting or egg dices. The time interval between two subsequent molting is known as stadium and the form assumed by the insect in any stadium is known as instar. Now we are going to discuss regarding Chawki rearing and late age rearing. Rearing of first and second instar larvae in silkworm is known as Chawki rearing. A rearing of third, fourth, and fifth instar larvae is known as late-age rearing. Temperature and humidity. Temperature and humidity play most important role during rearing of silkworm larvae. Silkworm do well in a temperature range of 23 to 28 degrees Celsius, with high temperature for the younger worms and lower temperature for the older worms. Humidity should be in the range of 70 to 85%, high for the younger worms and lower for the older worms. Temperature and humidity can be moderated by using wet gunny bags, wet newspapers, or pouring the water on cemented floors. In this table, optimum temperature and humidity for different instars larvae is given. For first instar, we have to provide somewhat higher temperature, that is 26 to 28 degrees Celsius. For fifth instar, we are providing somewhat lower temperature, that is 23 to 24 degrees Celsius. Similarly, higher humidity is provided in first instar and somewhat lower humidity is provided in the fifth instar. Silkworm density or spacing of worms. Different instars of silkworm requires different bed space. So spread this worm evenly and ensure that they do not overlap with each other. Overlapping or overcrowding leads to underfeeding, creating a microclimate for disease spread and could also lead to the suffocation. Here, silkworm rearing density and space is given in this table. For first instar, we have to provide 1 meter square area. For second instar, 2 meter square area. For third instar, 4 meter square area. For fourth instar, 8 meter square area. And for fifth instar, 17 meter square area. This is the rearing density of 1 case of silkworm. Now we are going to discuss stages of growth and feeding method in silkworm. In first stage or in first instar, the silkworm larvae are fed with young tender leaves, that is second and third leaves from the tip of the plant, and feeding is continued for 4 days. After this period, the worms go into the molt. Molting signs in silkworm larvae: swollen head, raised heads, worms are immobile. During molting, the bed should be kept dry and uncovered. Signs of getting out of molt: the worms are active and move around in the rearing bed. The mouthpart is broader. The body is dull with loose skin. Set up skin are easily seen on the bed. Once the worms come out of the mold, spread them out evenly and provide optimum space to match with their increasing body size. In second instar, feed the silkworm larvae with third and fourth young glossy leaf for 4 days. After this period, the worm go into the mold again. In third instar, feed the silkworm larvae with good leaves which are harvested from the green part of the plant, and feeding continued for 3 days. After this period, the worms go into the mold again. In fourth instar, feed the worms with the whole shoot and continue the feeding for 6 days. After this period, the worms go into the mold again. In fifth instar, feed the worm with whole shoot and feed for 7 to 8 days. During feeding, always maintain a single layer of shoot and avoid over mature and yellow and diseased leaves for provision of the food to the silkworm larvae. There are 3 methods of rearing there for late-age silkworms. They are self-rearing, floor rearing, and shoot rearing. See here self-rearing. These are the shelves over which bamboo trays are kept. Bamboo trays are more common in Karnataka and West Bengal, of diameter 1.2 to 1.4 meter. Self-rearing. In this method, silkworms are reared in bamboo trays, which are arranged one over the other in days on a rearing stands. Each rearing stand can accommodate 10 rearing trays. In this system of rearing, leaves are picked individually from the plant or branches of mulberry cut to the convenient size and fed to the silkworm at final instar. Usually, 4 to 5 feedings are given in a day and nets are used for cleaning the beds. This is the disadvantage associated with the self-rearing, and this is the advantages associated with the self-rearing. In self-rearing, cleaning is practiced more frequently, and also trays are removed from the stand for every feeding, so it is the labor intensive process and it costs somewhat higher. However, this method has the advantage of accommodating more silkworm in a limited space than any other methods. This method is preferred by the Indian farmers who are invariably confronted with the problem of rearing space. Now we are going to discuss regarding floor rearing. These are the fixed rearing seats of width is 1 to 1.5 meter. The space between two rearing seats is 0.6 to 0.8 meter. The length of rearing seat is 5 to 7 meter and it will be depend on the length of the rearing room. In floor rearing method, silkworms are reared on fixed rearing seats. The rearing seats are arranged in 2 or 3 tiers for accommodating large number of silkworm larvae. The rearing seats may measure 1 to 1.5 meter wide, 5 to 7 meter long according to the size of the rearing room. The space between the tay is 0.6 to 0.8 meter and sufficient space is left all around the rearing seat for attending to feeding and to carry out the other rearing operations. As in the self-rearing, the silkworms are fed with the leaves or branches which are cut into the small size. The number of feeding given here is 3 to 4 in a day. Bed cleaning is also followed in the same way as for self-rearing by using nets, but it is done less frequently here, twice during the fourth instar and thrice in the fifth instar. The main advantage in this system as compared to self-rearing is that feeding and cleaning process do not involve the handling of innumerable trays. As such, there is a considerable saving on labors. Last method is the suit rearing. This method is similar to that of floor rearing. Here also silkworm larvae are reared on fixed rearing seats, but which are provided with the whole suit. See here, we are providing whole suit as the feed to the larvae. Since the leaf supply due to suit feeding is distributed in 3 dimensions, there is better aeration of the rearing beds and therefore it is possible to have 50% more worms per unit area of rearing seat as compared to the self or floor rearing. Cleaning in this method also reduced to minimum. Cleaning in this method is also reduced to the minimum, which is normally carried out once each in the fourth and fifth instars. Process for bed cleaning. Ropes of convenient length are spread parallel to each other lengthwise on the rearing beds. Fresh branches of mulberry leaves are supplied for feeding over this row. After 2 or 3 feeds, when all the worms have crawled onto the new branches, the bed held by the rope is rolled into loose bundle and the old branches and dirt is removed. This is the most economical method of reading, as labor requirement for the process of feeding and bed cleaning are kept at the minimal level. In this table, leaf requirement for reading of 1 kg silkworm is given. In first instar, on first day we have to provide 150 gram leaves, on second day 200 gram leaves, on third day 200 gram leaves, on fourth day 150 gram leaves, on fifth day the larva go into the mold. Like that, in second instar, in third instar, in fourth instar and fifth instar we have to provide food according to this schedule. Bed cleaning. Bed cleaning is done to remove fecal matter, dead worms and leaf remnants which would promote fungal growth. It is done after every molt for the young silkworm, but it is done every other day for the mature worms. Mature worms require several bed cleanings. Bed cleaning is best done before feeding the worms. Here the procedure for bed cleaning is given. To clean the bed, spread a net over the worms and spread pressed leaves on the top of the net. Worms will crawl up to feed on the fresh leaves on the net. Transfer the worms using this net to the another clean bed or the tray. Remove any remaining worms which are left in the bed and then discard the waste into a compost pile. Thank you.